The overall goal of this video is to present and explain an efficient and easily adaptable method of fMRI in order to effectively explore the mapping of the human olfactory system without interference of natural breathing. One obstacle of using olfactory fMRI to study the brain activation responding to odorant stimulation is the inconsistency of repetitive odor stimulation, which is caused by the asynchrony of respiration and odor delivery. As illustrated here, when the odor presentation in a fixed timing odor stimulation paradigm is not synchronized with respiration, the repetitive odor stimulation can happen at different phases of respiration. A solution to this problem is to set up a respiration-triggered olfactory fMRI paradigm. Effective olfactory fMRI data collection is accomplished by first designing an event-related respiration-triggered olfactory stimulation paradigm on the olfactometer that is resistant to habituation and inconsistent inhalation patterns. The second step is to prepare odorants for their olfactory stimulation, taking care to use odorants that minimize trigeminal stimulation and habituation effects. The third step is to set up and run the experiment in the MRI environment by assembling the olfactometer with the capability of running a respiration-triggered paradigm. The final step is to process the data using the program onset to define the actual onset and duration vectors of the stimuli.
Ultimately, this technique has the potential to facilitate more sensitive exploration and connections of the human olfactory system.
Here we will demonstrate how to set up a respiration-triggered olfactory fMRI experiment using